So, good morning. I am thankful to founder and chairman of the Madhav University, Dr. Kumar Rana sir, chairperson Himmat Singh Deval sir, president Dr. Rajiv Mathur sir, organizer of this event, Dean Agriculture Dr. Geetam sir, and Dean Faculty of Basic and Applied Sciences Dr. DC Upadhyay sir, for giving me this opportunity in National Workshop on Recent Trends and Sciences with reference to the climatic change. So this is my topic, the phycomediation, a green nanotechnology approach for environment cleanup. So actually phyco nanotechnology, this is the amalgam of two things. One is the phycology, which means the study of the algae, and nanotechnology, that means we are studying the molecules of the size lesser than 100 nanometers. So this is the combination of two. So phyco nano remediation is the technology which is used to remediate many heavy metals, many pollutants and dyes, pesticides which are present in the industrial effluents discharged by many industries. Okay, so why we this is the background and why we need the phyco nano remediation, because there is an increasing industrial pollution and heavy metal contamination in the water bodies and the soils. So that's why we need the discharge the toxic pollutants like the heavy metals and the synthetic dyes and pesticides, hydrocarbons. So all these pollutants are being discharged in the water bodies and soil also. So that's why we need some technology. So there are many heavy metals like the lead, cadmium, mercury, chromium, arsenic. In fact, we are drinking those things also. So all these things were posing the risk. So effects of heavy metals on our body. So in fact, there are many vital organs affected by these heavy metals, like the neurotoxicity. So we can have the cognitive decline, that we cannot understand the things what's being spoken in front of us. So besides that, the neuroinflammation also, they may cause the neuroinflammation and the renal dysfunction. The filtration capacity of the kidney can be hampered, and respiratory issues, the inflammation and the reduced capacity of our lungs, and the cardiac problems also, like the arrhythmias. That means your heart will not properly beat, like the 72 beats per minute is required. So that may be less than that. So there are cardiac risks of the heavy metals, and the oxidative stress. So because of the oxidative stress, there can be the formation of the free radicals which will kill your cells, and the immune dysregulation. So there are two types of cytokines, pro-inflammatory cytokines and the anti-inflammatory cytokines. So if the level of the pro-inflammatory cytokines is elevated, there is a huge inflammation in our body, and in fact it is the initiation of the cancer. So there are cytokines, the level of them is elevated, like the TNF alpha and IL-11, IL-6 and INF-7, interferon gamma, and on the other hand there are the anti-inflammatory cytokines which are helpful. But because of these heavy metals, the level of those helpful cytokines is decreased. So multiple effects of these heavy metals. So why do we need this technology, phyco nano remediation? Because of there are the limitations of the conventional remediation techniques. There are many, however, there are many techniques like the chemical precipitation, ion exchange, electrochemical treatment, and membrane filtration. These are the techniques by which with which we can remove the heavy metals. But they have some problems because they don't have the sustainability and the high operational requirement, maintenance cost is high, and the insufficient at low concentration. That means the concentration of the heavy metal in your material in the water body is too less. Those techniques are none of use. Okay. So rising interest in the green nanotechnology solutions. So there are the biological systems, in fact, like the algae, bacteria, fungi, and plants. So these we can use for the remediation. But I will focus on the phyco-nanoremediation, that is the study of the algae. And with those algae, we will try to synthesize the nanoparticles. And with those nanoparticles, we will remove the heavy metals in the contaminated water bodies. Okay. So how do we biosynthesis the nanoparticles? So this is the simple diagrammatic representation of the biosynthesis of nanoparticles. We add the metal salts of the metals and in the algal biomass, and algal biomass have so many bioactive compounds which can reduce these ionic species of the nanoparticles. So ultimately we will form the nanoparticles and we will purify the water. So there are many ways to synthesize the nanoparticles with the algae, that is a phycosynthesis, brown algae mediated biosynthesis. So these are the algae with which we can synthesize the nanoparticles. First in the Terminaria conites, this nanoparticles, it can produce the silver nanoparticles, and the synthesis conditions will be the 1 hour incubation in the dark. So we have to create the dark condition for the synthesis of nanoparticles with this particular algae, Terminaria conites, and the spherical size of those nanoparticles will be of the 96 nanometer. And the characterization, once we synthesize those nanoparticles, we have to characterize them whether those nanoparticles are useful or not. If the size of those nanoparticles is too much, the agglomeration rate is too high, so there is no use of those nanoparticles. So that's why we have to characterize those nanoparticles with the many sophisticated techniques. Many sophisticated techniques like this scanning electron microscope, transmission electron microscope, Fourier transform, infrared, and the XRD. These are the techniques with which we can characterize the nanoparticles. And what is the thing inside this algae which can reduce the ionic species of the nanoparticles, so we can have the metallic form of those nanoparticles. So these are the reducing agents, and in this Terminaria conites is the carbonyl groups and the polyamines which can reduce the ionic species of the nanoparticles. Another is the sargassum species of algae. This species can produce the silver, not the silver but the gold nanoparticles. Okay, and the location is the extracellular. That means the silver nanoparticles are being formed outside of the cell. That means they are releasing some polyamines or the reducing agent which can reduce the ionic species of the nanoparticles. And the synthesis condition, there is the requirement of the synthesis, pH 5 and incubation at room temperature. So the room temperature 25 degrees Celsius will be sufficient for the synthesis of these nanoparticles. And the longest ages, hexagonal and trigonal shaped nanoparticles will be formed, and which will be in the range of 300 to 400 nanometer. And characterization of those nanoparticles, we can use the AFM, XRD, FTIR, and UV-Ris, and the reducing agent present in this algae, hydroxyl and amine functional groups. Another is the Sargassum muticum, with which we can produce the zinc oxide nanoparticles, and these zinc oxides also are produced outside of the cell, so the location is the extracellular. So synthesis condition, heating at it's very high temperature at the 450 degrees Celsius. So you can imagine we can die, of course, at that high temperature. At this high temperature, the zinc oxides nanoparticles are produced by the Sargassum muticum. And the shape, hexagonal shape nanoparticles will be formed in the size of the 30 to 57 nanometers. And the characterization of those nanoparticles can be done with the UV-based SAM, FESCM, FTIR, and the XRD. And the reducing agent, that means with those reducing agents present in the Sargassum muticum, which will reduce the ionic species of the metallic particles are the sulfate and hydroxyl groups. And next is the Aclonia cava, that is also the algae, and it can produce this gold nanoparticles. The location of that also is the extracellular, and the synthesis condition 1 minute incubation at the 80 degrees Celsius in the magnetic stirrer. So we need the magnetic stirrer for this process, because for many reactions they are the stirring requirement. Okay. So spherical size of the nanoparticles will be obtained by the Aclonia cava, and we can characterize the nanoparticles with the UV base FESM, TAM, XRD, and FIR, and these are the hydroxyl and phenolic groups which will reduce the ionic species of the nanoparticles. Next is the Terminaria conoids also, but in this case that is also algae, which I have already mentioned earlier, but with this time we can produce the gold nanoparticles instead of the silver nanoparticles. So Terminaria conoids, extracellular location, the requirement of the synthesis 1 hour for continuous stirring at room temperature. Room temperature is sufficient, and the characterization of these gold nanoparticles can be done by the EDS, that is the energy dispersive X-ray spectroscopy, and the SEM. And these are the biochemical groups, reducing agents. And next is the Aclonia cava. So with the Aclonia cava algae we can produce the gold nanoparticles also, and the silver nanoparticles as well. So the location is extracellular, 1 minute for 80 degrees Celsius. That means the time is too less for the synthesis. That is a plus point for the synthesis of the gold nanoparticles with Aclonia cava. And the spherical nanoparticles will be produced, and the FCC, the face-centered cubic nanoparticles will be produced. And the triangular shape of the nanoparticles also can be obtained with the size of the 20 to 50 nanometers. And the characterization of this gold nanoparticles can be done with the FTIR, FESM, EDS, and UV vis, and these are the reducing agents, amines and hydroxyl groups, which are ultimately responsible for the synthesis of the nanoparticles. Okay. So next is the Padenia gymnospora, that is also algae, and we will produce the silver nanoparticles with this Padenia gymnospora. Extracellular is condition, and the temperature range is 24 to 95 degrees Celsius. Spherical nanoparticles will be produced, and the characterization of this gold nanoparticles can be done with AFM, UV vis XRD, and HRTM. These are the very sophisticated technology with which all of you will face in your future while you will be writing the research papers, okay, or some articles or book, whatever you are doing. These sophisticated equipments you will require, and many of them are available in our university, so this is the matter of pride. So these are the fucoxanthin and flavonoids, the reducing agents, and next is the Sargassum muticum, with which we can produce the silver gold nanoparticles. The location is the extracellular, and the synthesis condition 1 hour stirring at 45 degrees Celsius. And the FCC spherical nanoparticles will be produced of the range of 6 nanometer, and zeta potential and UV vis TAM can be used for the characterization. So I included one more equipment, zeta potential. So if there is more minus or more plus, that means there is the less amount of the agglomeration of those nanoparticles. Otherwise it is close to zero, that means there is more agglomeration, there is larger molecules of the nanoparticles, which is none of use. So these are the proteins and bioorganic compounds as the reducing agents present in the Sargassum muticum. So till now I described the brown algae. Now I come to the red algae mediated biosynthesis. So red algae which are mainly present in the sea. So Gracilaria adulis, this is the red algae with which we can synthesize the silver nanoparticle. The location is extracellular, and the synthesis condition incubation on orbital shaker and 150 rpm. So many times we can provide the rpm, so those media will rotate only in that condition the silver nanoparticles will be produced. So these are the conditions which have to be fulfilled by us, otherwise the nanoparticles might be synthesized, but those will be of larger size. Okay. So size is spherical and 12 to 100 nanometer characterization, UV vis, SAM, TEM, XRD and FTIR, and these are the proteins and terpenoids. So these terpenoids are also present in the plants and the algae, which have many medicinal importance. So next is the Xylidium MNC, the silver nanoparticle extracellular, and the synthesis condition 48-hour incubation at room temperature, and the centrifugation at the 13,000 rpm. So you can imagine what is a 30,000, how fast it will rotate. Okay, so too much rotation, faster rotation. Shape and size, the spherical size and characterization UV vis, and next is the Kappa ficus Algal-Mediated, with which we can produce the gold nanoparticles. The location is extracellular, and the size and shape, that's the polydispersed. That means that your sample will have the nanoparticles of different sizes. And characterization of that can be done XRD, TEM, FTIR, FAS and UV vis. These are the very sophisticated equipments, and as the reducing agent you can use phenolic compounds. So these algae use the phenolic compound to reduce the any species of the nanoparticles and converting it in the metallic form of the nanoparticles at last. So next is the Galaxy-Ura elongate, that also is used for the synthesis of gold nanoparticles. The location is extracellular, and the incubation 10 minutes to 12 hours, so incubation period is too much. So at continuous stirring at 120 rpm, so there are incubator shaker also with which we can set that particular rpm, that is the rotation per minute. So how many time something rotates per minute. So that is the RPM. Okay. And with the Galaxy-Ura elongate, we will have the rod truncated and triangular shaped nanoparticles in the range 3.85 to 77.13 nanometer, and the characterization of those nanoparticles with Galaxy-Ura elongate, the zeta potential, TEM, HPLC and GC, and this is the palmitic acid present in the Galaxy-Ura elongate, which will reduce the ionic species of the nanoparticles. And the Chondrus crispus, another algae with which we can synthesize this gold nanoparticles. The location is the extracellular, and room temperature is sufficient for this process, but stirring is required, and pH will be between 2.4 and 10. Okay. The spherical and polyhedral shaped nanoparticles can be synthesized with Chondrus crispus or in the range of 30 to 50 nanometers. So in most of cases, the size of the nanoparticles should be lesser than 100 nanometers. If it is greater than 100 nanometers, so it is no more nanoparticles. Okay. So next is the Lamania fluviatilis. That is very difficult to pronounce algae, and the synthesis of this gold nanoparticles can be done with this algae. The location is the extracellular, and the room temperature stirring is sufficient for the synthesis of this gold nanoparticles with this Lamania. And the polydispersed FCC face centric cubic structures and the spherical structures will be produced in the size range of 5 to 15 nanometer, and the characterization of those nanoparticles can be done with the UV based XRD, FTIR and SAM. So next is the Corallina officinalis. Okay, this is another algae. So with which we can produce the gold nanoparticles. The extracellular is location. That means the nanoparticles are produced extracellularly. So FCC and spherical shaped nanoparticles will be produced, and characterization can be done with UV vis, SAM, XRD, FTIR and TEM. So these are very sophisticated. In fact, the SAM or TEM one equipment costs more than 1 crore. And there are other equipments also like the NMR, which cost more than 4 crores. Okay. So next is the Geladiella acerosa, with which we can produce the silver nanoparticle. The location is extracellular, and the 48 hours incubation in with the 120 RPM will be sufficient, with which we can obtain the FCC and spherical particles, and the characterization UV vis, SAM, XRD, FTIR, TEM. So these are the aromatic compounds. Nanoparticles can be synthesized chemically, biologically and physically also. So all these streams are able to synthesize the nanoparticles, but the means are different, the ways are different with which nanoparticles are produced. So in this way, this is cost effective, eco-friendly, because we are using the organisms, algae, which is not going to harm us in any way. So that's why this technology is preferred to the conventional technologies. So next is the Acanthophora spicifera, with which we can produce the silver nanoparticles. The location is extracellular. 20 minutes of incubation will be sufficient for the synthesis of these nanoparticles at 60 degrees Celsius. And spherical 48 nanometer sized nanoparticles will be produced, and the characterization techniques are the TEM, FTIR and UV vis, and these are the alcoholic, carboxylic. These are the alcoholic, carboxylic and phenolic compounds which act as reducing agent for the reducing of the ionic species of the metals. And there are the blue-green algae also. We can use, we have used the brown algae and the red algae. Now I come to the blue-green algae, in which the cyanobacteria are also involved. So this is the nostoc ellipsosporum. So that is algae, and that is blue-green algae. Okay. So with which we can synthesize the gold nanoparticles. The location will be extracellular, and the shape and size decahedral and the icosahedral. So there are many shape, decahedral, icosahedral, hexagonal. You have already studied in your 11th and 12th classes if you are a student of science. So synthesis condition, 3-hour incubation and PS5. Characterization of the gold nanoparticles with the nostoc algae can be done with the UV vis, SAM and FTIR, and there are the reducing agent, proteins and carboxylate groups. So in fact, we can functionalize the nanoparticles also. We add in cold blood these functional groups, so that nanoparticles can perform in a better way. So next is the Spirulina platensis. In fact, Spirulina platensis, however, we have studied that algae, that means these are the multicellular organisms, but this is not the case. In fact, Spirulina is the prokaryote, and you must have known what is the difference between the prokaryotes and eukaryotes. So there are many exceptions you will study in the biosciences and agriculture sciences. Next is the Spirulina plantasis, with which silver gold nanoparticles, but the conditions, if the conditions are changed, the agglomeration rate and shape and size, all things will be obtained in that way. So this is the important factor, that what conditions you are setting for the biosynthesis of nanoparticles. So Spirulina platensis is spherical size 20 to 30 nanometers, and the synthesis condition 40-hour incubation is required. That is very long, and the characterization of these gold nanoparticles synthesized by the Spirulina platensis can be done with the UV phase, FTIR, TEM, SAM, EDX, XRD. So these are the techniques. And as reducing agents, these Spirulina platensis use biomolecules like the amino, carboxylate, phosphate, thiol. So all these groups could be used for the reduction of the ionic species of the metallic nanoparticles. Next is the Spirulina platensis, and next is Plactonema boreanum. बोर हो रहे हो क्या भाई? Plactonema boreanum, please keep silence. Plactonema boreanum, this is another algae with which we can synthesize the gold nanoparticles. The location is extracellular, and the size of nanoparticles will be in the range of 20 to 25 nanometers, and the synthesis conditions 24-hour incubation at 200 degrees Celsius. That means for this synthesis there is the very high temperature is required, 200 degrees Celsius, and the characterization can be done with the TAM, XPS, TOF, that is a time of flight, TOF, mass. So we can connect many equipments also like GCMS. So we can connect those equipments. So one task will be done with one equipment, and another task will be transferred to the next equipment. So that's why there is a combination of many equipments also many a times. So these are the next is the Kushrestha species with which we can produce gold nanoparticles. The location will be extracellular. Spherical sized nanoparticles can be produced in the range of 3 to 13 nanometer. So with very smaller nanoparticles can be produced, but we have to keep in mind if it is lesser than the 10 nanometer, it can harm the human body also. So we have to decide a range perfectly. There are the fine lines at two corners. So 60 hour incubation is required at 20 degrees Celsius in light, and the TEM, SERS and zeta potential are the characterization techniques for the synthesized gold nanoparticles by the Kushrestha species, that is algae, and carboxylic groups and polysaccharides are the reducing agents found in this algae. So next is the Limbia majuscula and Spirulina subsalsa. So gold nanoparticles, the synthesis is intracellular. So here you can see a difference between other algae and this algae. So this algae is producing the nanoparticles intracellularly, while I have mentioned all the algae which were producing the nanoparticles extracellularly. So with this Limbia majuscula and Spirulina subsalsa, we can synthesize the nanoparticles intracellularly. But anyhow, we have to take those nanoparticles out of the algae. So then only we can use those nanoparticles for the purification of the contaminated bodies. Okay, so Formedium tenu, this is another algae which can synthesize the silver nanoparticles extracellularly. Spherical and irregular shaped nanoparticles can be produced in the range of the 14 to 16. And these are the techniques. And here are the enzyme and metabolites. Because enzymes are not released out of the cell. There can be the conditions, but most of the times enzymes work inside the cell. So that's why these enzymes can act as reducing agent in this condition and can reduce the ionic species of the metallic nanoparticles. That's why. So in this case, enzymes and metabolites are acting as reducing agents. In other cases, biomolecules, phenolics and flavonoids, which are also released outside of the cells. So next is the formadium species that produce the gold nanoparticle intracellularly. Triangular shaped nanoparticles will be produced. The incubation at room temperature and the characterization of these nanoparticles can be done with UV-Vis HRSEM. HRSEM is a more sophisticated equipment, that is a high resolution scanning transmission electronoscope. Okay, and FTIR and EDX. So these are the very sophisticated techniques with which the characterization of the nanoparticles can be done. Okay, so in this formadium species, these are the cytoplasmic proteins which can synthesize the nanoparticles. So in every case there are some differences. In some cases the differences of location, in some cases the differences of type of nanoparticles. There might be different synthesis conditions, there might be different characterization processes, and the most importantly there might be different reducing agents with which we can synthesize different kinds of nanoparticles. So characterization of those nanoparticles. So these are the shape and the algae with we use. The brown alga, red alga, and blue-green alga, and micro alga, macro alga, with which we can produce this polymeric ANPs or the metallic ANPs. There are the metallic oxides also, like I just took one instance, zinc oxide. So in the oxide form also the nanoparticles can be produced. The porous silica, this form of the nanoparticles also can be used, and the nanorods and the mesospores and the functionalized ANPs. In functionalized ANPs we add the functional group to those nanoparticles to make them more effective. So these were the techniques which I have already mentioned in my earlier slides. So mechanism, these are the mechanisms with which these nanoparticles can remove the heavy metals, or in the water, contaminated water bodies, the heavy metals being discharged from the industries as industrial effluents. So bioaccumulation. So in bioaccumulation, this is a bioaccumulation. So this is the algal cell wall. So this is the single wall, single algal cell wall. Algae are multicellular organism. Okay, this is the single wall in which I am describing the mechanisms adopted by this algae. So there could be the bioaccumulation of the heavy metals inside the cells. There could be the degradation also. So those nanoparticles may cause the degradation of this nanoparticles also. There could be the absorption. So those those heavy metals will directly enter inside the cell of the algae. But before that they will complex with nanoparticles. Then they will be absorbed inside the cell, and there could be the biotransformation also. That means those heavy metals will be produced in insoluble forms. So those can be sedimentation by some techniques like the centrifugation. So these are the mechanism with which the nanoparticles synthesized by the algae can be used for the removal of the heavy metals in the water bodies. Okay. So application in the pollution removal. So the nanoparticles produced by the phycoremediation. There are some application like the heavy metal removal. They can remove the PB, lead, cadmium, chromium, and mercury, and dye degradation also, methylene blue and rhodamine B, and wastewater treatment. The industrial effluents also can be treated with the nanoparticles, and the CO2 sequestration and biosensing. These are the advanced techniques we can use. So these are the application in the removal. Advantages of the phycoremediation. So these are the eco-friendly and cost-effective techniques. Okay, so that's why these techniques are preferred as compared to the other physical hazardous or other techniques. And renewal biomass source, because the algae will be reproduced with time. So we don't need to add the algae again and again, because they are living organism and can reproduce on their own. You know this thing already, what is the difference between living and non-living thing. And sustainable nanomaterial production can be done, and the potential for circular bioeconomy, that means the things will be used again and again. So these are the advantages of the phyco-nanoremediation, but definitely everything has some limitations. So there is the scale of difficulties. That means, so when we apply these things at a very large scale, we can have the scale of difficulties, and the nanotoxicity also concerns, because those nanoparticles can be harmful to your body also. That's why you have to create the nanoparticles of a particular size only. And environmental safety and regulatory issues and need for mechanistic studies and field trials are required. So these are some limitations. The future prospects of the phyco-nanoremediations is the genetic engineering of algae for enhanced nanoparticle yield. So you know everything is the genes. Okay, in this world, these are the genes, because of which whatever you are doing, whatever you are, at last genes are responsible for that thing. Okay, so if we manipulate those genes with the genetic engineering, we can produce the algae with the desired characteristics. The algae with those characteristics which will help us produce the higher amount of nanoparticles, the more effective nanoparticles. So these are the future prospects, and there can be the integration of the artificial intelligence for the process optimization, and the future prospect large scale wastewater treatment and the biosensing applications. References. And this is my publication published in the Springer Nature with the Dr. Anupama Srivastava, ITMVU Vadodara. So thank you so much for listening me so patiently. Thank you so much.